The following program is brought to you by Caltech.
Good evening. I'm Elliot Meyerowitz. I'm chairman of the Division of Biology, and I'm here to introduce Dr. Siapis. But first, let me announce that there will be a special Watson lecture on July the 7th, in which Ed Stone, who's familiar to all of us and of great importance at the Jet Propulsion Lab for many reasons, will give a special lecture as part of NASA's Summer of Innovation outreach to middle school students. But we think the talk will be of interest not only to middle school students, but to everybody, and you're all invited. Now for the speaker. Athanasios Siapis has been on the Caltech faculty since the first day of 2002. And in that time, he's earned tenure and his current title of Associate Professor of Computation and Neural Systems. He has a joint appointment in the divisions of Biology and the Division of Engineering and Applied Science. As you'll guess from his name, he was born and grew up in Greece. And the quiz question is, the town is where the 8th Ecumenical Synod was held in 869. I've got $1 for anybody who can tell me where Thanos was born. His undergraduate and graduate degrees are from MIT. He obtained his Bachelor of Science in Theoretical Mathematics and Electrical Science and Engineering in 1990, and his PhD in Electrical Engineering and Computer Science. His interest then shifted to neurobiology, and he did postdoctoral work, again at MIT, with a professor there, Matt Wilson, who is a Caltech graduate, and in the MIT Department of Cognitive and Brain and Cognitive Sciences. There, Thanos began the development of his now classic methods of multi-electrode recording from live and behaving animals, which has allowed an unprecedented level of information about the activity of nerve cells in the brain to be collected and analyzed, while the animals, which are rats, eat, sleep, wander, and find places in their living quarters, and even while they dream. Don't ask me what they dream about. Ask Thanos. Thanos is also integral to Caltech in ways other than his research. He's a resident associate in Avery House, for example, and so he's an important part of our system of undergraduate houses. But today it will be the rats and not the undergraduates that he'll be talking to us about. Thanos?
Thank you. Thank you very much, Elliot, for the introduction. It's a real pleasure to have the opportunity to tell you a little bit about the process of memory formation and also about some of the work that we have been doing to try to understand the brain circuits that are involved in this process. So the general question we would like to ask is, how is it that the brain acquires and stores new information? And as I will describe, this is a very complicated process that requires the coordinated activation of multiple brain areas, and it takes a very long time to complete. And part of the reason is that the brain is not simply trying to store the memory like you would store a file in a hard drive, but it actively tries to integrate it with all previously stored items of information. So when you retrieve a memory, it automatically comes with all the right associations. And this is the property that makes the brain such a powerful computing machine. So making progress in understanding this, how the brain does this can have interesting implications in a couple of different domains. First of all, it may change the way we build machines that process and store information. And second of all, understanding how the normal brain stores memories may give us new insights as to how what goes wrong during diseases of memory, such as Alzheimer's disease, and may accelerate the search for cures for such diseases. So, so how does the brain do this process? So the short answer is we don't know. Well, there are some things that we do know, and so I'll begin by telling you a little bit about what are the facts that we do know about the process of memory formation. Then I will tell you about some of the open challenges and some of the approaches that we and others are trying to use to make progress in this problem. And finally, I'll give you a couple of examples of work that we have been doing to understand some of these brain circuits that are involved in memory. So if we are to, so if we ask in what part of the body are memories actually stored, the safe bet is that all the action is taking place in the brain. And here you can see the brains of several animals. And as you can see, there is a lot of variance in the sizes and the complexity of these brains. However, a lot of them share very common architecture. So if we, let's say, for example, cut in half a human and a rat brain and look at them from the side, you can see there are several key brain areas that I'll be discussing later on. This green region that is basically at the surface of the brain is the cortex. You can see it in the rat here. And there are several key subcortical areas, such as the hippocampus, that we'll discuss in more detail. So the key elements that compose these brains are cells that are called neurons. And as you can see here, that they have a wide variety of shapes and properties, and they are connected into precisely organized circuits. But despite this great diversity in shapes, as you can see, there is a universal language of computation and communication in the brain. And in particular, most of these neurons communicate with each other with electrical pulses. So when a cell's membrane increases beyond a certain point, it sends out a pulse that another cell receives, and then its membrane voltage increases. So if we were to record the membrane voltage of a cell, what we would actually see as a function of time is something like this. And then these sudden deflections that you see that last less than a millisecond, less than a thousandth of a second, these are the electrical pulses that the cells send to each other to communicate. So, and these are called spikes. So this is an artist's rendering of this process. So these neurons have parts of these processes called dendrites where they receive the information. And then when the cell body increases in voltage beyond a certain point, they produce these electrical pulses that travel down the axon, the cable that is the output of the cell, and then influence other cells. And the way they influence other cells is through these specialized interfaces that we call synapses. And here's a drawing of one of the synapses, and this is the membrane of the cell that receives the input, the postsynaptic cell. And when the electrical pulse goes through the synapse, it releases neurotransmitters, molecules that, through a sequence of steps, change the voltage in the postsynaptic cell. So the amount by which the voltage changes is related to the strength of the synapse. And different synapses in the brain can have very different strengths. So for example, you can have very weak synapses, such as the one from A to C, that give a small change in the voltage of the cell, or very strong synapses, such as this one, that can produce a bigger change in the voltage. And the synaptic strength is not fixed, but changes over time. And one of the key factors that control how this synaptic strength changes is the timing between spikes, the timing between inputs and outputs to the cell. So many experiments have shown that when a cell, such as this one, receives an input right before it spikes, then that causes an increase in the corresponding synaptic strength. And conversely, if the cell receives an input right after it spikes, then the synaptic strength decreases. So the timing of inputs is very important for controlling how the different synaptic strengths are distributed in the brain. And furthermore, it also shapes the type of activity that we see in the brain. So if you have these different cells that fire in an uncoordinated fashion, their influences on the cell would not add up. And typically, a cell receives input from a few to a few thousand cells, and input from a single cell is not enough to make it fire. you require coordinated inputs from a lot of cells that their influences would add up and cause further spiking and further propagation of activity. So the general questions that we would like to ask is whether there are general principles that we can identify that determine how electrical activity flows through the different circuits of the brain. And second of all, how is it that patterned activity that flows through these circuits changes the synaptic strengths in the network? So given this framework, I'd like to tell you a little bit about some of the history of how the basic ideas concerning memory formation have evolved. So the first question we would like to ask is, where in the brain are memories actually stored? Here's a picture of a human brain, and the cortex is composed of many different areas that specialize in different functions. So there are parts of the brain that process visual information, other ones that process auditory information, other parts that are responsible for language comprehension or generation. So, and this idea that the function in the brain is localized started early in the 18th century. So this guy, Franz Gall, had the notion that different parts of people's personalities would be in different parts of the brain. And he thought that if he fondles people's skulls, he could tell how much cultured they are or whatever. So obviously his ideas ended up being completely wrong, but they had a seed of truth. Indeed, there is localization of function in the brain. And in that spirit, people thought that memories, there must be a part of the brain that actually stores memories, sort of like a hard drive of the brain. So early in the 20th century, Carl Lashley, a psychologist at Harvard, decided to try to figure out where such a brain region, if it exists, and where it would be. So what he did is he trained rats to navigate mazes and remember how to do that. And then he systematically lesioned different parts of their cortex, and he tried to figure out whether there is a part of the cortex whose damage would have a disproportional effect in memory. So that would be a candidate of the memory storage area. So after a series of experiments, he could not identify such a region, and he summarized his conclusions with these two principles. So the memory in particular was reduced in proportion to the amount of damage in the cortex, and all areas of the cortex seem to be sort of equally important for learning. So that got people to start thinking that memories must be distributed must be very distributed throughout different cortical areas. But the question became, if this is true, then what would be the substrate of these memories, and how could you make such a distributed representation? So Donald Hebb, a Canadian psychologist, make suggestions of how both of these problems could be solved. And he suggested that memories must be stored in the pattern of synaptic connections across a distributed population. And he suggested also a simple rule by which these synaptic connections would systematically change to create these memories. So in particular, he said that if a cell systematically contributes to the firing of another cell, then the corresponding synapse would be strengthened. And this is the rule that I talked about earlier, but Hebb proposed this in the absence of, way before we had any experimental data. So it was a very insightful idea that had a very big influence in research. So how did he think we would create memories out of this rule? So imagine you have a very distributed network of cells. Because of the way sensory inputs come, you would get systematic activation of a group of cells. This would cause the corresponding synapses to strengthen, and this would glue this collection of cells together. So for example, as you listen to what I say, parts of your auditory cortex may be activated, and then parts of your visual cortex may be activated by the colors on the slide. And the synaptic rules would glue all these neurons together to form a coherent whole. So is that really true? And if so, how exactly would things work out? This has been a very influential hypothesis that has driven a lot of the research, as I will describe. So the next really major breakthrough in our understanding of the process of memory formation came from a very famous clinical case, the case of patient HM, which you can see here. This is a picture around the time where he started to have very strong epileptic seizures that could not be controlled with drugs. So a neurosurgeon in Connecticut, William Scoville, decided to try to take out a part of the brain known as the hippocampus that you can see here in order to try to control these epileptic seizures. There was no other way to do it. And the reason that this area is called the hippocampus is shown here. As you can see, there is a remarkable similarity to the seahorse, and hippocampus is the Greek word for seahorse. So after the surgery, HM had profound memory deficits that were very carefully characterized by a Canadian researcher, Brenda Milner. In particular, she found that while HM could remember things from his childhood, very old memories, it was impossible for him to create new memories. So there was a researcher that worked with him for many years, and he still could not recognize her. So he would meet her every day in the morning, and it would be as if they met for the first time. So maybe hippocampal damage is the secret to a happy marriage. So, and this, of course, created a lot of, there are a lot of movies that have been made patterned after HM. How many people have seen this one? Yeah, so HM is like the guy in this movie, only he didn't go around killing people. And there are lighter versions of the same story, basically. So this was a really remarkable finding that changed the course of memory research. Here was this strange brain area, the hippocampus, that was important for making new memories, but did not actually store the old memories. And the hippocampus is, from an evolutionary point of view, a very ancient part of the brain that is, whose basic architecture is very much preserved across species, as you can see. So this led to a whole series of experiments in other species where people try to understand the participation of this structure in memory. And indeed, just for example, in rats, and you can see the hippocampus of a rat in a diagram of the rat brain here, all the phenomena that have been observed with clinical patients have been replicated and extended, and in particular, rats that have a hippocampal damage can no longer form new memories after the time of the hippocampal damage, but they remember their rat childhood, and the memories before the time of hippocampal damage are affected in proportion to their recency, with very distant memories unaffected, as I mentioned. So this led to two conclusions. First of all, since you can no longer make new memories without the hippocampus, this is a critical machine for making new memories in the brain. But since you can remember very old memories, the memories must be stored somewhere else. So where are they actually stored? Well, we don't really know, but the predominant conjecture is that they must be stored in a very distributed fashion throughout neocortical areas. And this process of memory that is referred to as memory consolidation extends over very long periods of time. So we know that even experiences that can last even 20 seconds can trigger reorganization of brain circuits that can extend over months in rats and years in humans. And during these periods where the brain gets activated in a very heterogeneous ways. So in particular, there is a drastic difference in the activity patterns that we have during awake behavior and during sleep. So this led to the hypothesis that maybe these two different brain states have a complementary role in the process of memory formation. So the general idea is that during awake behavior, external inputs drive cortical areas, which in turn somehow drive the formation of a memory trace within the hippocampus. And then during sleep, the hippocampus becomes spontaneously active by its own, and somehow drives the consolidation of memories in cortex. But is that really true, and how would this happen mechanistically? So there's very little that we know in detail about this process, and the reason is that, as you can see, it requires monitoring the activity of many cells, both in the hippocampus and also across cortical areas, in freely behaving animals and for long periods of time. And these type of experiments have been technically very difficult and have only recently been possible. So for the next few slides, I'll tell you a little bit about some of the techniques that we try to use to monitor activity over long periods of time. And first of all, part of the challenge in understanding circuits like the hippocampus is that the way information is represented is very distributed. And so here's an example of population coding from real life. This is a bunch of University of Washington fans cheering for Caltech in the 1961 Rose Bowl. Of course, needless to say, Caltech did not play in that game. So each of these fans has no idea what they're doing. In fact, they think that they're spelling out something different. So information doesn't exist in any individual card, but only across the relationships across many of these cards. And similarly, to really understand the information across these circuits, it only exists in the relationships of the activity across multiple cells. So how can we try to track the activity across a big population of cells? So the classic technique that has been used in the past is that of extracellular microelectrodes. So these are very fine tip electrodes that are advanced very close to the cell body, and then you can eavesdrop on the cells and listen to the electrical pulses that they send out. There is one possible confound with this approach, which is if the electrode tip is equidistant from two different cells, it may get the same signal, and it would be hard to tell which cell is firing this spike. So, for example, if we wanted if someone was heckling me from the audience and there was a single microphone to figure out where the voice is coming from, it would not be possible to do it. But you can have multiple microphones that can be used to triangulate the location of the sound source. So similarly, people have made electrodes that have multiple recording sites, such as this one. This is called a tetrode because it has 4 recording sites that you can see here. And it's a bundle of 4 microwires that are twisted together into a single recording probe. And the basic idea is very simple. Because of the spatial separation between the tips of the wires and the signal sources, each time each of these cells spikes, these spikes will be picked up on all 4 wires, but with different amplitudes that depend on the location of the cell with respect to the wires. So for example, when the blue cell fires, the highest amplitude will be on the green channel because it's closest to it, and the lowest will be on the yellow channels because it's furthest from it. And when the red neuron fires, it will be highest on the 4th channel and the lowest on channel 1. So if you try to compare the amplitudes, to plot the amplitudes on channel 2 versus 3, you may not be able to tell these 2 signal sources apart. But if you plot the amplitude on channel 1 versus 4, they should split up into clearly isolatable clusters. So this is how things should work in theory. How do they work in practice? So here's examples from the hippocampus. As you can see, spikes are picked up on all 4 channels, but with different amplitudes. And you can use this information to isolate the activity of multiple cells in the vicinity of the tetrode tip through a process of analogous to triangulation. And furthermore, when these recordings take place, you not only see these electrical pulses, but also very clear oscillations in the extracellular field, the local field potential, that I will describe in more detail later. So we can design arrays that allow the independent adjustment of multiple of these electrodes, and that we can use to record from multiple brain areas simultaneously. And then we can monitor the activity as the animals perform memory tasks. They try to remember locations in space, or the fact that certain tones predict rewards, and so on. So I want to give you a sense of what this activity actually looks like. So I'll show you a short movie where you can see basically 2 very striking properties of electrophysiological activity in the hippocampus. So first of all, these are recordings from 4 different electrodes, and here you can see the spikes on one of these electrodes. So the first striking property is that this circuit continuously oscillates very regularly, as I mentioned before. These are known as theta oscillations, and they are 4 to 10 hertz in frequency and very strongly modulate or clock neuronal activity in this network. And the second amazing property is that cells tend to fire in very specific locations in space, and hence the name place cells. So the location of the rat will be indicated by these 3 lights that you see here. And you'll see that as he runs on this linear environment, this cell will only fire here. And basically, you will listen to each of these spikes as they hit the speaker, as they fall off the left-hand side of the screen. So you can see that this circuit continuously oscillates, and this cell only fires at this location in space, and it fires very rhythmically. And this happens over and over and over. It's a very robust property of this network. So in contrast, when the rat sleeps, and in particular in a stage of sleep known as slow wave sleep, these regular oscillations go away, and instead what we get is very synchronous bursts that are interspersed with irregular activity. So this will be basically this synchronous burst that you will hear when they hit the speaker. So it's random activity interspersed with these very synchronous bursts. In contrast, in another stage of sleep, REM sleep, which is when dreams are believed to, I guess that's what rats are dreaming of, then the activity looks, again, these rhythmic oscillations come back, and activity looks similar to awake behavior in that sense. So there are these two very different modes of activation of this network that are illustrated here. So you can see this is the activity of about 130 or so simultaneously recorded cells. Each row represents the activity of a single cell, and each tick mark is one of these electrical pulses. So what you can see is that the way the population is activated changes drastically from this point to this point. And this is, in fact, an episode of REM sleep that is sandwiched between two slow wave sleep episodes. So these two major modes of activation of the network have mirror images in awake behavior. When the rat runs around, the activity looks very much like this, theta modulated. And when he stops to drink water or just hang out, then his brain switches and he tends to produce these very synchronous bursts. So for the last part, I want to tell you a little bit about how we try to understand how these networks operate. And in particular, we've seen that spike timing is a very critical property that controls both the plasticity of synapses in the networks and also just the way activity flows through it. So we'd like to understand how is activity organized throughout the hippocampus in awake behavior. How is the timing controlled during the performance of memory tasks and awake behavior. And as we've seen before, during active processing of the environments, the hippocampus oscillates very regularly, and we can think of the phase of these oscillations as a little clock, so that will rotate 360 degrees when it hits one full cycle. And you can see that these cells don't fire uniformly across time, but tend to fire around 3 o'clock. So in that sense, this oscillation clocks hippocampal activity and structures how this network is activated. And not only that, these theta oscillations are very important for controlling plasticity in this network. So many experiments have shown that if you deliver pulses at 100 milliseconds, that is theta frequency, this is a very it's a powerful way of inducing plasticity in these networks. And finally, this clocking signal may be very important in coordinating the interactions of the hippocampus with other brain areas. So this is, for example, some experiments that we have done a few years ago, where we showed that this area of the cortex, known as prefrontal cortex, it has about 40% of the cells in this area are very strongly modulated by the hippocampal theta oscillations. So you can see an example here. These are the hippocampal theta oscillations, and this is a cortical cell that tends to fire close to the peak of these oscillations. So it becomes important to understand how these oscillations are organized throughout the whole hippocampus. So here's a three-dimensional model of the hippocampus. You can see the cortex shown in yellow here. And this red area is a part of the hippocampus known as area CA1. And I'll focus most of the discussion on this area for the next few slides. So as we've seen, we can think of these oscillations as little clocks that modulate activity in the network. And the hypothesis for a long time is that these clocks are perfectly synchronized. and they remain synchronized over time and produce very correlated output from the whole structure to whatever areas receive input from it. And so we wanted to test this hypothesis and understand in more detail how these oscillations are organized. And to do that, we can topographically map activity in these circuits, and we can demonstrate that indeed the electrodes are sampling the network activity in a grid-like fashion. You can see in these MRI images, you can see it actually more clearly here, the grid emerging at some depth. So we wanted, so for the next few slides, I'll show you a top-down view of this array, which is going to look something like this, and we're going to compare these oscillations across different parts of the hippocampus. So, as is well known, when the rat is actively exploring the environment, the whole circuit oscillates, and you can see indeed oscillations recorded throughout this array. And here I have arranged these traces so that as we go down each of these columns, the corresponding traces are arranged in sequence like this. So if we were to look, zoom in this 1 second segment and look at these same traces as an image, you can see that there are very clear phase differences, and this sort of violates this hypothesis of synchronous activation. And furthermore, these phase differences are very systematic. You can see these diagonal lines as we go down each of these columns of this array. And so the fact that the phases are not the same, but they change very systematically, tells us that these oscillations must not be synchronous, but there must actually be waves that travel in this direction across the hippocampus. So we can explicitly test this hypothesis, and we can do that by making movies of the instantaneous phases across the whole array and analyzing these movies. So just to contrast with the previous hypothesis, if these oscillations were synchronous, we would see the whole array going up and down with the same phase. But in what we actually observe is this. So you actually see these very clear propagating waves that sweep through the hippocampus. So this suggests that this network is activated in a very different way than we originally thought, that there are these waves that sweep through the whole structure, and this changes the way we can think about how targets of this structure receive information, and opens a lot of interesting directions. So for one thing, it tells us that there is no single clock in this whole network, but time in the hippocampus is organized just like time on Earth, like in a regular progression of time zones. And there are many questions that have to do with what are the mechanisms that cause these waves to propagate, how do they get modulated by behavior and learning, and how do they affect the processing of information in these networks. So I'll tell you very briefly about what we think this means for the coding of information in this network. So as we saw, there are these two very striking properties of activity in the hippocampus. It oscillates in theta, and then different cells tend to fire in very specific locations in space, and these are called place cells, and the corresponding locations in space are called place fields. And John O'Keefe, that has discovered place cells, has shown that these two properties are related in the following sense. So as the rat goes through its place field, this corresponding cell fires at earlier and earlier phases of the theta rhythm. So this is a cartoon of the same notion. So imagine that the rat goes through the place field. Initially, the cell will fire here, and then a little earlier, and earlier, and earlier, and earlier. And if you have a cell that starts firing at this point, it will also do the same thing. But this has a very important implication that, as you can see, cell 1 will fire before cell 2 on every single theta cycle. So if we consider a third cell and a fourth cell, you will see that cells that have overlapping place fields will not all fire together, but they will fire in sequence, 1, 2, 3, 4, that reflects the relative position of the place field centers. So how does this relate to the presence of these waves, and what does this mean? So we can do a thought experiment. Imagine that we could indeed look through the whole hippocampus, and we could freeze time and see what cells would be active when the rat, let's say, is in this location in space. And of course, it would be cells that have place fields that overlap the location of the rat. And many experiments have shown that these cells are scrambled throughout the whole hippocampus. There's no systematic map. They're just randomly distributed throughout the hippocampus. But as I mentioned before, these cells will not be all activated at once, but they will be activated in sequence. So let's say when the phase is at this point, only the cells that, the A cells would fire. And if the hippocampus is synchronous, if these oscillations are synchronous, and we could freeze time, we could see only A cells firing at this instant in time. And if the phase advances, then only the B cells would fire, and then only the C cells would fire, and so on. So if at any instant in time, the total output of the hippocampus would represent a single point in physical space. So how does this view change when we have these traveling waves? Well, under these conditions, you would see all the phases represented in the hippocampus in a systematic fashion. And instead of a point, you would get a segment A, B, C, D. And the point of discontinuity, AD, would travel down the same direction as these waves. So this suggests that maybe these segments of space are the elementary units of hippocampal computation. We can try to ask, why would this be the case and what are the advantages? I'll tell you one possibility. So imagine that this is the trajectory of a rat in a two-dimensional environment, and we wanted to encode the sequence of points that the rat follows. If we would try to do that with a sequence of points, then we'd have a problem when we try to recall this sequence, because when we reach this red point, we don't know if it's part of this branch or of this branch. But if we represent it as a sequence of segments that appear to be the instantaneous output of the hippocampus, this problem goes away, and we can represent these sequences as non-repeating patterns. So, of course, this opens a lot of questions that have to do with why is it that this structure is activated in this strange way? How does this correspond to, how does this participate in memory formation? And there are a lot of experiments that are underway to try to elucidate some of these questions. So for the last part of the talk, I want to tell you a little bit about how activity during sleep is organized. And so we know that sleep is very prevalent in the animal kingdom. Many animals spend a big part of their lives in this state. Must have come at a very high evolutionary cost, because animals are vulnerable to predators in this state, and you don't seem to be doing anything useful. But the fact that it's so prevalent, it must mean that it must play a very important functional role. So there have been several hypotheses of why we sleep. It's fair to say that it's still an open question. We don't really know what the functional role of sleep is. But it's clear that the brain remains very active and also very plastic in sleep. So as we've seen, it gets activated in a very structured way. So this must have very systematic consequences in the way these networks get interconnected and the connections change in sleep. So one of the hypotheses has been that because during sleep you turn off the inputs to the brain, it becomes an opportunity to remodel it in such a way so as to consolidate memories. And this is a hypothesis that sort of remains an open question. But I'll tell you a little bit about some work that we have done to try to understand one aspect of sleep activity, which is this very synchronous population burst that we see in the hippocampus during sleep. And so these are spectacular population events. About 40,000 cells fire together in less than 100 milliseconds during each of these bursts in CA1. And these bursts happen about once a second or so. So the question is, what are the consequences of these bursts for cortical areas and also for the hippocampus itself? So I'll first tell you a little bit about what we think these bursts do to the hippocampus, and then I'll describe some of the influences of these bursts to cortical areas. So let me begin by sort of a general observation that if you have a network of cells, the strength of the connections in this network would control the degree of order in the network. So this is easy to see in the two extremes. If you have very weak coupling, each of these cells is not really influenced by its neighbors. It doesn't pay attention to any other cell, and it does whatever it wants. This is sort of the Greek model of interactions. So therefore, the activity in this network will become is uncorrelated. And in contrast, when the coupling is very strong, then the network produces very correlated output. But in practice, these connections do not have fixed strengths, but they're constantly changing under the influence of these plasticity rules that I previously described. So we have this feedback where the network activity would control the synaptic strengths, which will control the network activity, and so on. So it was widely believed that synchrony should increase synaptic strengths. But according to this picture, if synchrony increases synaptic strengths, this would increase synchrony, which would increase synaptic strengths, and so on, and there would be a problem. So I'll try to argue that the exact opposite happens in certain networks, that is, synchronous activity actually leads to weakening of the synapses, and in a very selective way. So why is this the case? So first of all, as I mentioned, one of the main factors that control the changes in synaptic strength is the timing between inputs and outputs to cells. So if a cell receives inputs right before it fires, then that would lead to strengthening of the corresponding synapse. And conversely, if it fires and then receives input, then that would lead to weakening of the synapse. And as you can see, this means that very small differences in timing can have very big consequences, very opposite consequences for the organization of these networks. And one of the key factors that controls the timing between inputs and outputs of these cells is the presence of axonal of these delays. So we wanted to ask what would happen when you have delays and synchrony in a network that is plastic. So we illustrate the main idea in this very simple, the simplest possible network, which is two cells that are connected to each other. If these cells fire at the same time, then you'll see that they will receive each other's input after a short delay. So every time they fire, they will receive each other's input. And if you receive input right after you fire, this would lead to weakening of the connections, according to what we said, right? So this is the basic idea. Two cells fire, they will receive each other's input. That should lead to weakening of their connections. And we have studied this property a lot, both theoretically and experimentally. And you can see that, furthermore, this weakening will be selective. Only the neurons that fire together at the same time will have their synapses weakened, because it's only these cells that receive each other's input right after they fire. So if you have a complicated network like that and you wanted to selectively weaken the connections between a very specific subset, this need not have a very simple biophysical implementation, but this idea suggests a very simple solution. If you just make the cells fire together, that should take care of it. So I want to tell you a little bit about how we think this selective erasure caused by synchrony may be involved in this process of memory formation. So imagine that we have a cortical network and then a hippocampal network here. So during awake behavior, patterns of external inputs will systematically drive some cortical cells, which in turn will drive some hippocampal cells. And if this happens systematically, as we've seen, then we would get progressive strengthening of the connections between this specific subset of hippocampal cells. And the hippocampus is a very plastic structure, so it captures this type of temporal relationships very, very easily. And below, I show a representation that it's convenient to think about the patterns stored in these networks as wells in a landscape, with how deep and wide the well is corresponding to how strong the corresponding pattern that is stored there is. So the question is, what happens during sleep? So during sleep, there is no external input. The external input to the hippocampus is attenuated, and this network becomes spontaneously active. And when this happens, the cells that are very strongly connected to each other will have a much higher chance of being activated in these bursts. And as I mentioned, if this is true, then every time they get activated, they will selectively weaken the connections between them. They will erase the corresponding trace. And so the strength of the pattern that we store acts as a counter that gets decremented each time this network, this population gets activated, and controls the number of times that the pattern is allowed to be expressed. And you can see that it won't completely erase the pattern, but it will weakening up to the point where it's sort of similar to the strength of other patterns stored in the network. And this is important if you want to put in a new pattern and have a chance to let it play out. And so the question is, what do we expect the corresponding changes in cortex to be? So as this pattern expresses and progressively erases itself, we would expect a corresponding pattern to reinforce itself in cortex. Is that really true, and how would we test this? It turns out to be a very difficult problem because these changes are expected to be gradual and also distributed across the network. So we don't know the answer, but I'll tell you a little bit about some work that we've tried to do to make some progress in this direction. So the first question is, where in cortex would you look? And we decided to look in this area here, this prefrontal cortical areas. And one of the main reasons is that this is an area that is directly connected to the hippocampus. So if there is hope to figure out how cortical and hippocampal networks are activated together, this is a very good candidate to try to do that. So a while ago, we've shown that these synchronous bursts in the hippocampus usually happen together with slower oscillatory periods in cortex known as spindle waves that last about 4 seconds, 1 to 4 seconds or so. And so we wanted to ask, well, is it when this activity happens in sleep, are the precise timing relationships between these networks? We've seen that these are very important for causing synaptic changes. So that's the first question. And then the second question is, well, if there are such precise timing relationships, are they the same across sleep, or do they change depending on, let's say, REM versus slow wave sleep? So I'll very briefly mention the bottom line and then tell you the general picture that emerges. So this shows basically the tendency of a single cortical cell to fire in relation to a bunch of simultaneously recorded hippocampal cells. So each of this row is a pair of that single prefrontal cell with a hippocampal cell. And whenever you see red, this means that this single prefrontal cell have a strong tendency to fire right after this specific subset of hippocampal cells. So whenever the cell fires, it tends to fire right after a very specific subset of hippocampal cells. And so the question was, well, in other stages such as REM, would we expect the same timing relationships or maybe reversed? But we're very surprised to see that basically all correlated activity seems to go away, and during REM, these two networks appear to function sort of independently in an uncorrelated function. So the general picture that emerges is that whenever you have this very synchronous burst in the hippocampus, this causes a very specific subset of cortical cells to be activated. That depends on which cells in the hippocampus are activated. And when these bursts are very strong, this causes a secondary wave in cortex that makes a different set of cells firing about 100 milliseconds later. So when the hippocampus kicks the cortex with these bursts, it causes these waves to appear. So obviously there are a lot of open questions that we and others are trying to explore that have to do with what controls what cells fire in this burst and what controls what corresponding cortical cells firing with them. Is this related to the previous experience of the animal? Does this change with learning and so on? And a lot of the challenges have to do with developing techniques that allow us to really monitor the activity throughout the process of memory consolidation. So we would like to really take a very simple memory from the point that, from the first experience of the animal, and really track it throughout this whole maturation process. And there's a lot of work currently done in this direction and trying to see what patterns we expect to see. So let me summarize some of the key points in the talk. So first of all, I mentioned that there's a brain structure, the hippocampus, that plays a critical role in memory formation. But this structure actually doesn't store the long-term memories. It just helps establish them somewhere else, and most likely across distributed cortical areas. And we've seen that the timing between spikes across these networks is very important for controlling both the changes in synapses and also the flow of activity in these networks. So we did some work to try to characterize how this timing is organized both in awake behavior and sleep. And I've shown you some evidence that the hippocampus is controlled by these traveling waves of activity, and that there is these waves also influence cortical areas, in particular these prefrontal cortical areas. And finally, during sleep, the communication between the hippocampus and this cortical area seemed to be exclusively structured around these very discrete bursts. So this suggests that these are the key events that may drive the consolidation of memories in cortex. So let me thank the people that actually did the work. So it always helps to work with people that are much smarter than you, and this is definitely the case here. So a lot of the work that I described, the traveling waves in the hippocampus, and also this decoupling of synchrony leading to decoupling in the hippocampus was done by this amazingly talented postdoc, Eugene Lubanov. And Kaz is another fantastic postdoc that has worked on this prefrontal hippocampal communication. And Ming, Andreas, Laurent, and Maria, just fantastic people in the lab that I didn't have a chance to describe their work, but hopefully at a future point in time. Thank you.
This program is brought to you by Caltech. Visit us at caltech.edu.